Hello, everyone. I'm Sylvia Lee and want to welcome you back to the Diatom Web Academy, organized by the Diatom Taxonomic Certification Committee of the Society for Freshwater Science and diatoms.org. You can follow us on Facebook, Twitter, YouTube, and join our email list for the latest updates. I have a couple announcements before the webinar. Have you ever wondered where you can buy some diatoms? There are at least 4 sources of diatom cultures to check out. You can find out more about them at the link in the chat in the news article by Sarah Spalding. And the second announcement I have is you only have to wait 1 week from today for the next Web Academy. On Tuesday, November 16, Nick Schulte from University of Colorado Boulder will present about the application of diatom multimetric indices to state monitoring data. So be sure to tune in next week or catch the recording. So with that, I'll hand it over to our speaker today.
Thank you, Sylvia, and good morning, everyone. My name is Isabel Hong, and I am an NSF Earth Sciences postdoctoral fellow, currently located at Simon Fraser University in British Columbia, Canada, but also co-located at Central Washington University. I am broadly interested in understanding coastal hazards by using modern day observations to help inform our understanding of past natural hazards, such as earthquakes, tsunamis, and storms. I got my bachelor's degree in geology from Whitman College in Walla Walla, Washington. I then got my master's degree also in geology from Central Washington University. And then I attended Rutgers University, where I received my PhD in oceanography. And it was at Rutgers where I got into studying diatoms and learning how they can be used as a proxy for reconstructing relative sea level change. And so it is this work that I'm excited to share with all of you today. I would like to begin by acknowledging that this work that you'll see here today was done on the ancestral, traditional, and contemporary lands of the Coast Salish, the Confederated Tribes of the Chehalis Reservation, as well as the Chinook Indian Nation, in particular, the lands of the Lower Chinook tribe, as well as the Willapa tribe, who since time immemorial have inhabited these lands. However, this recognition only becomes meaningful when combined with accountable relationships and informed actions. And so this acts only as a first step in honoring the land upon which this research was conducted. And so this brings me to talking about my study area and why there's interest in understanding the relative sea level history of Willapa Bay. Historical and instrumental observations are often insufficient in understanding past relative sea level, because tide gauges and historical observations haven't been around long enough for us to really understand what that long-term record of sea level history can look like. This can also be further problematic in areas where tectonic processes can be a primary driver of local sea level. One such place is Willapa Bay, which is located along the active Cascadia subduction zone. And Cascadia has a record of repeated great earthquakes that cause instantaneous relative sea level rise, or if we were to think about it from a land record perspective, instantaneous land level subsidence. And we only know this due to evidence preserved in the geologic record. Now, because the last great earthquake happened over 300 years ago in 1700, we don't have a complete understanding of these coastal hazards without investigating the geologic record. So then to understand the potential risks associated with relative sea level rise, or conversely, land level subsidence from these great Cascadia subduction zone earthquakes, we have to really look at investigating the coastal sediments preserved in low energy depositional environments that will allow for the preservation of this land level change as changes both in the coastal sediment record, but also in the fossil diatom record. And so this led to my overall study motivation, which is to see how we can develop diatoms as a proxy that will allow us to reconstruct land level change during these past Cascadia subduction zone earthquakes. So to start, I'll talk about how diatoms can play such a crucial role in these types of reconstruction studies. Diatoms are incredibly useful in coastal land level reconstruction studies because they're abundantly found in marine, brackish, and freshwater environments. Additionally, their frustrals preserve really well. They're pretty resilient in preserving themselves in the geologic record, so we can use them then and the frustrals to identify them to species level, and then use their known salinity preferences to reconstruct paleo elevation with respect to the tidal frame. So how does this work? First, we'll start with looking at this cross section here that you see. This is a cross section of an idealized salt marsh setting where we have the subtidal environment on the bottom right. Here, I can bring my mouse around, and then we have the upland environment in the upper left. So that as we increase in elevation, we are decreasing in salinity. And so we're going to see that relationship reflected in the diatom species that we find, where if we were to stand in the subtidal environment and look at the diatom species there, what we would find are predominantly marine diatoms. But as we continue up in elevation, moving into the tidal flat and then into the low marsh environment, we would begin to see a shift away from marine diatoms towards brackish diatoms. And then as we move from the low marsh into the high marsh and upland setting, we'll see a shift happening that's moving away from brackish diatoms towards freshwater diatoms. And so if we take a step back and look at this diagram as a whole, what we would notice is that there's a vertical zonation of these diatom assemblages with respect to the tidal frame. So then what we can do is we can make similar inferences to the same species that we find in the fossil record to then reconstruct paleo elevation over time. And so my talk is going to follow two general broad stories. The first story I'll talk about today is in the creation of this regional modern diatom database, where we seek to understand the modern distribution of diatom species in tidal wetland environments throughout Willapa Bay, and then to see how different environmental variables are affecting the diatom distributions that we're observing in the present day. The second story then will be focusing on how we can then use those modern diatom distributions to then develop and apply a transfer function to reconstruct land level change caused by these Great Cascadia subduction zone earthquakes. So my study area, as I mentioned, is located in Willapa Bay. Willapa Bay is boxed for you and labeled as B in map A. As I mentioned, Willapa Bay is located along the Cascadia subduction zone. I have Willapa Bay shown for you then in more detail in map B with accompanying bathymetry. So these warmer colors indicating shallower depths and these cooler colors indicating deeper depths. And Willapa Bay itself is really large. It's about 350 square kilometers, and about 2/3 of Willapa Bay itself falls within the intertidal. Now, tides of Willapa Bay are mixed semi-diurnal, with the Great Diurnal Range actually ranging from 2.8 up in the north and to about 3.3 meters down to the south. You might also notice here in map B, I'll show it for you here with this mouse, there's also a bay here. Now this 26-kilometer spit actually protects Willapa Bay from the Pacific Ocean, and that's resulting in allowing for these low energy wetland environments that will allow for the development and accumulation and preservation of sediments and fossil diatoms that's really conducive for reconstructing past sea level. And furthermore, that much of Willapa Bay is nationally or state protected, meaning that they're relatively undisturbed. So it would make for then a really great location to study the relationship between diatoms and their environment, and then to then try to make similar inferences to reconstruction studies. So then sampling took place in May of 2015, as well as August of 2019, during which average daily temperatures and mean precipitation were similar to historical averages. In total, we ended up sampling 7 shore perpendicular transects from 4 tidal wetland environments, totaling 104 samples. Now, the 4 tidal wetland environments are shown for you with the black circles in map B. So moving from north to south, we have the Smith Creek, Bone River, the Newaycom River, and the Nacelle River, with each of those locations shown for you in more detail in maps then C through F, and the locations of the sampling transects. Now, we sampled more than one transect at some of these locations in order to capture a wider range of environments that we wouldn't have been able to do from just a single transect. At each of these transects and from each of the sampling stations within each of these transects, we also measured elevation using an RTK GPS or a total station, and then tie those measurements to a tidal benchmark that allowed us to relate these sampling locations relative to mean tide level. In addition to collecting samples for diatom analysis, we also did pore water salinity measurements, collected samples to take back to the lab and do grain size measurements, and we also collected samples to look at total organic carbon. Now, for diatom analysis, I counted 400 valves and identified and classified the species based on where possible local observations, and if not possible, then using global observations. For diatom analysis, in addition to counting the species present, I also did hierarchical clustering to identify separate groups of diatom assemblages within each transect. And then I followed that up with principal component analysis and redundancy analysis to analyze the relationship between the modern diatom assemblages and the environmental variables. And so sampling, as I mentioned, was done along shore perpendicular transects. And the goal of doing that then was to sample from multiple intertidal zones, ranging then ideally from the subtidal up into the upland environment. As I mentioned, sometimes that wasn't possible from just a single transect. And so we did our best to collect multiple transects and try our best to capture a wide array of environments. And so sampling happened with every 10 to 20 centimeter change in elevation. Where that wasn't then possible were places where there were these steep slopes geomorphologically that prevented us from capturing every 10 to 20 centimeter change in elevation, in which case we just did our best to capture that change. So then ideally what we would see in our sampling then in sampling in the subtidal environment, here you see a picture of us out in a canoe using a grab sampler to try and capture all the diatoms that we could find in the subtidal environment. And then sampling in the tidal flat involved getting waders on and doing our best to wade out as low as possible and far out as possible to collect samples. The low marsh environment, where we began to see more presence of vascular vegetation. And then the high marsh, you can see that the vegetation becomes much more dense, and we sampled there as well. And then in the upland environment, forgot to take a picture of sampling in the upland, but this picture does a very nice job, I think, of showing you the transition of the upper limit of the high marsh environment as we transition into these forested upland environments. And so the sampling would then continue past that transition and then up into the upland environment. So I'll talk about the results from one of the transects. Here I have the results from Bone River Transect 1, this B to B prime. where I have increasing distance inland on the x-axis, and then the environmental variables that we measured on the corresponding y-axes. And so I'll start at the top of the environmental variables, focusing on the elevation profiles. So here we find that as we move inland, elevation increases. And based on the vascular vegetation that we saw while we were sampling, we divided our sampling transect here into 4 intertidal zones: the tidal flat, the low marsh, the high marsh, and the upland environment. Below the elevation profile, I have plotted for you results of the grain size analysis, here shown for you as a percentage of mud. So percentage of mud as opposed to coarser grain materials such as sand. And what we find there is that as we continue inland, we find that the percentage of mud-grained sediments increased. Conversely, as we moved inland, we found that salinity decreased. And as we moved inland, we found that organic content in the form of total organic carbon tended to increase as we moved inland. And then for the diatom results, I have plotted for you above each of these sampling stations the diatom species in percent relative abundance. And the diatom species that you see plotted here had to have an abundance of at least 5% in one of the samples. And then I classified them for you based on their salinity preferences. Again, local observations were possible, and if not, then using global observations of their salinity preferences. And what we found is that the diatoms showed some type of zonation here at Bone River Transect 1. And what I mean by that is I'll focus here first in the tidal flat environment, where the diatom species that we find present are marine in their preference. And that as we enter into the low marsh environment here, we begin to see the presence of brackish diatoms. And then finally, as we enter and transition into the high marsh environment is where we begin to see the presence of freshwater diatoms, or diatoms with a brackish freshwater preference. And so we wanted to kind of evaluate this relationship a little bit further. And we did that using, as I mentioned, hierarchical clustering, principal component analysis, and redundancy analysis. And the summary results of that I've shown for you here on this sample environment biplot, where axis 1 is the axis of greatest variability and axis 2 is the axis of second most variability. And what we find with the sampling stations and clustering analysis is that clustering analysis identified 3 distinct clusters along this transect. With the green cluster, we find sampling stations 1 through 6, which comprise of the tidal flat environment. The second cluster then comprises, the purple cluster comprises sampling stations 7 through 11, where we see low marsh samples. And then that brown cluster comprises of all of the sampling stations from the high marsh and the upland environment. To then look at how the measured environmental variables seem to play a role in driving that diatom variability, that's what you see plotted here as vectors. So looking at the environmental vector that most closely parallels axis 1, which is our axis of greatest variability, we find that it's elevation. And so here at Bone River Transect 1, we see that elevation appears to be the primary driver of the diatom variability that we're seeing here in this transect. And so to characterize for you generally what we saw with the individual transects and combining all of those observations into looking at characterizing it from a regional scale, what we found is that in the subtidal and the tidal flat environment, we found generally coarser grain sizes. So they tended to be sand-sized grains as opposed to silt or clay-sized grains. That generally in the subtidal and the tidal flat environment, we found low to no organics, and salinity was really high. As we transitioned into the low marsh environment, we tended to see that grain size decreased, became more fine, the amount of organic materials tended to increase, and that salinity began to decrease. And then moving and transitioning into the high marsh and upland environment, where we tended to find there were fine grain fine-grained sediments as well as higher organic contents. And there is where we found low to no salinity measurements there. And then characterizing the results of looking at the diatoms, we found, again, so from combining all of the results, we had a total of 104 samples in which we identified 300 diatom species across 85 genera. And clustering analysis was a little bit more complicated in this sense. So what we found with clustering analysis is that we ultimately identified 3 distinct clusters. Cluster B, here shown as the purple cluster, tended to have captured the higher elevation samples, whereas cluster A, the green cluster here, tended to have captured the lower elevation samples, and then cluster C captured then sort of the middle elevation samples. But what we found is that there was a lot of overlap in the sampling stations relative to elevation with these clusters. And then the results of the sample environment biplot that you see here, with each of the symbols indicating a single sampling transect, is that we found that a lot of these environmental variables seemed to closely parallel axis 1, with actually grain size here shown for you as percentage of mud was the environmental vector that most closely paralleled axis 1. But as I noted in the previous slide, because we tended to find this sort of co-varying relationship between elevation and these environmental variables, where we saw an increase in finer grained sediments as we increased in elevation, and similarly we saw an increase in organic content with elevation, and then conversely a decrease in salinity as we increased in elevation, we find that the vectors all tended to pull in parallel with each other along this axis 1, because elevations seem to display then this co-varying relationship with all of the other environmental variables. And so our takeaway was that in reality, elevation, even though it wasn't the primary driver here, grain size seemed to be the primary driver of diatom variability on a regional scale, we were able to show that elevation was a primary driver of this variability. And so I wanted to then take a sort of different look at the relationship between diatoms and elevation. And that's where I did weighted averaging. So I did weighted averaging to determine the elevation optima and tolerance of the diatom species. So looking at the elevation at which each diatom species was most abundant, and then by calculating the tolerance, looking at the range of elevations in which we continue to see that diatom species present. And so the results of that weighted averaging is shown for you here on this slide, with the most abundant species being that they had to have relative abundance of greater than 5%. So that ended up actually being only 65 species. And I've plotted them for you, labeled them for you, again, based on their salinity preferences. Here, red for freshwater, yellow for brackish, and blue for marine. And then on the x-axis here, I have for you elevation in relation to the tidal frame as this standardized water level index, SWILI. And that in doing that, that allowed for comparing these samples that were taken from different tidal ranges. And so as I mentioned, the tidal ranges range from about 2.8 to 3.3 in Willapa Bay. And so to standardize that, we use this SWILI index. And as a point of reference for you, I have the second x-axis on the bottom here. So mean lower low water, for example, is equal to 0 SWILI, mean tide level is equal to 100 SWILI, and mean higher high water is equal to 200 SWILI. And what we found with the optima of these diatom species is that the elevation optima ranged from below mean tide level to above mean higher high water, and that the tolerances were then much greater than that. And so looking then at species and indicator species to talk about ways in which we could sort of characterize these different intertidal zones, what we then saw was that species with the most narrow elevation tolerances tended to be then species observed to have more of a freshwater preference, such as Cosmonaeus pusilla, Ludicola mutica, Nicha terrestris, and Pinularia lagerstedtii. And these tended to then be re-identified species that would be indicative of these high marsh or upland environments. We also tended to observe then species such as Gyrosigma eximium and Mastigloia exigua as species that had relatively narrow tolerances and could then be used as indicator species of low marsh environments. In the tidal flat, species with sort of narrower tolerances were Chromatophora oceanica and Opophora pacifica. But as I mentioned here in this lower bullet point is that what we did find is that compared to freshwater diatom species that tended to have very narrow elevation tolerances, marine and brackish diatom species tended to have much wider elevation tolerances. And actually what we found in addition to that is allochthonous species or species with allochthonous components, for example, Coconaeus scutellum or Paralius leucata and Planethidium delicatulum were observed across multiple environments. And so moving forward with potential sea level reconstructions at Willapa Bay, there's some species there that we have to think about that would be perhaps less useful and less indicative of intertidal zones that we would maybe want to consider pulling or removing from our reconstructions moving forward. And so I wanted to then shift a little bit and then talk about sort of the story 2 of developing and applying a diatom transfer function to reconstruct relative sea level using these modern observations. So as I mentioned at the beginning of this talk, instrumental and historical observations are really insufficient in understanding and capturing that long-term variability that we hope to have to really understand what those sea level change risks can look like, especially in places with a long and active subduction zone. But what's great is that we can use estimates from microfossils, for example, diatoms, preserved in the geologic record to then produce more accurate measurements of relative sea level rise through time. And this has wider implications beyond just measuring sea level at a single point. By having these sort of estimates of land level change and using a transfer function allows us to produce sample specific errors as well. What that allows then geophysicists to do is to take those estimates to then model and understand the likely variations that we're seeing in co-seismic slip distribution. And an example of what I mean by this is this figure here shown. So study done by Wong et al. And the authors of the study compiled all existing transfer function estimates of land level change from the most recent Cascadia subduction zone earthquake from 1700 with the estimates of that land level subsidence shown for you adjacent on this map to 1 sigma uncertainty. And just based on those transfer function estimates, they were able to model the likely rupture patches from that 1700 earthquake, showing that actually the rupture wasn't just this clean entire rupture that was uniform across the entire subduction zone, that there was actually some variability along that subduction zone. And so that led to sort of wanting to develop a diatom transfer function that I could apply to the Cascadia subduction zone to provide estimates of not only the 1700 earthquake, but from older earthquakes to really see what that variability can look like. And so how do diatoms really play a role in all of this? I wanted to then talk a little bit about how diatoms can play a role in the tectonic setting that we're seeing in the Cascadia subduction zone. And that involves talking about a concept known as the earthquake deformation cycle. And what I have for you here is this block diagram showing you the tectonic setting where we have the subduction of the Juan de Fuca plate underneath the North American plate. And that's creating this stuck zone that's causing very gradual uplift in coastal environments. And that gradual uplift is allowing for the establishment of salt marshes and tidal wetland forests that's allowing for the development and accumulation of soils in which we would find predominantly freshwater and brackish diatoms. If we were to measure land level during this interseismic period that is present day, where we're in a time preceding a Great Cascadia subduction zone earthquake, what we would then find is land level will be gradually rising over time. But as we move into the co-seismic period, that stuck zone becomes unstuck and the plates are freely moving, and that's going to cause instantaneous subsidence onshore. Oftentimes the release of that energy can be really great, and it can generate then a tsunami that will deposit a tsunami sand onshore. Because that tsunami sand is eroding and transporting and depositing sediments from offshore to onshore, what we find in that sediment is a mixture of diatom assemblages ranging from marine to freshwater. And if we were to measure land level change during this co-seismic period, what we would find is a very sudden and dramatic drop in elevation. But as we re-enter into the interseismic phase, because we dropped in elevation so dramatically during this co-seismic period, these coastal environments are now sitting at a much lower elevation. And so they're going to have an accumulation of more intertidal muds, in which we would find predominantly marine and brackish diatoms. But as that unstuck zone becomes stuck once again, we're going to have gradual uplift occurring in these coastal environments. And again, that's going to allow for the reestablishment of salt marshes and tidal wetland forests, where we'll see a transition happening then from intertidal muds into more organic rich soils. And that change is also going to be reflected in the diatoms, where we see a shift from marine and brackish diatoms back into freshwater diatoms. And so what we can do is we can take those qualitative observations that we're seeing both in the sediments, but also with the diatoms, and make that fully quantitative, making those elevation measurements with those sample specific errors using a method known as a transfer function. And so with transfer function, then the goal of re-investigating a site like Willapa Bay is that, as I mentioned, Willapa Bay is advantageous for a modern diatom study, but it's also really advantageous for a fossil diatom study like this, because there have been a lot of scientists and paleoseismologists who have gone to Willapa Bay to find this sort of stratigraphic geologic sediment record of these mud over soil contexts I just described, that's evidence for this land level change that we're seeing caused by Great Cascadia subduction zone earthquakes. And what was great about this study here referenced by Atwater and Hemphill-Haley in 1997 is that Eileen Hemphill-Haley actually looked at the fossil diatom assemblages and used a method known as factor analysis to show that we're seeing changes in these diatom assemblages that are indicative of a great amount of substance through time. And then, so the goal was to revisit one of these locations that was described by Brian Atwater and Eileen Hemphill-Haley to then collect a sediment core where we can see that change happening in land level preserved in the geologic record. And so we collected a core from Willapa Bay, where you can see here on the picture of the core opened in the laboratory, where we see that that really dark band is that soil that's dropped in elevation, and we see the accumulation then of these intertidal muds above that. And so really targeting these types of contacts to then look at the fossil diatom assemblages that we see. In addition to looking at the diatoms, we also measured in the laboratory grain size analysis, and also by looking at the organic content in the form of doing loss on ignition. So taking all these sort of laboratory measurements, in addition to looking at the fossil diatoms, to piece together that reconstruction of paleo environment and land level change through time. And so how do we develop then a transfer function? What does that look like? And so the first step is in quantifying what's happening with the modern day distribution. And that's what I talked about previously in this talk, where we see different diatom species and their relative abundances across the tidal wetland environment. And the next step, similar to what I described for you with doing weighted averaging, using the transfer function to then identify the optima, the elevation optima, and the elevation tolerance of these diatom species. What the transfer function then does is it creates these species response curves that you see here colored to show how each species is responding to our environmental variable of interest, and in this case, elevation. What we do then is we look at the fossil diatom assemblages in the sediment core, and the transfer function then is able to look at those fossil diatom assemblages and then recreate paleo elevation, as you see on the far right here in panel C, with these sample-specific errors. And then from there, look at that change in elevation across that contact to measure the amount of subsidence from these earthquakes. Now, the challenges that we've been facing with developing a diatom transfer function to reconstruct land level change in Cascadia is this modern analog problem caused by the high species diversity of diatoms. And what that looks like then is that we can have diatom species. So for example, here in panel B, a species F that's really abundant in the fossil diatom record that we don't see in high enough abundance in the modern day distribution. And so what that results in then is the transfer function is unable to provide a reliable estimate of paleo elevation. And from there, we can't estimate the amount of land level change through time. And that was a problem that I faced with my study. And so after working with a great colleague of mine, Neve Cahill at Maynooth University, who's a Bayesian statistician, we came up with a solution to group diatom species. And that's what you see here in panel A. And so by looking at similarities in diatom species abundance and distribution across elevation, actually grouping these diatom species to create here what you see in panel B, then group response curves to then ultimately use those to inform the transfer function to then reconstruct land level change. And so in doing that, then it's required us to sort of pivot a little bit and to create this new transfer function. And our new transfer function is this Bayesian method. And by going with a Bayesian route and developing a new Bayesian transfer function, it allowed us to actually incorporate these flexible group response curves. And so instead of having to rely on a really Gaussian distribution with a singular optima and tolerance, what we were then able to do by incorporating this Bayesian approach is to actually have multimodal response curves and to actually try to then capture the reality of the distributions that we were seeing with the modern diatom data set. Now, what makes the transfer function Bayesian is the ability to add informative priors. And so here we use lithologic priors to help improve our probabilities and to sort of make the paleo elevation reconstructions more into a formally defined range. So what I mean by that is if I were sampling in the sediment core from a unit I knew and could identify as intertidal mud, we knew that that intertidal mud could not have formed any higher than mean higher high water. So what that means is that the mud couldn't have accumulated any higher than the upper limits of the high marsh environment. Conversely, if I were sampling in the sediment core from what I knew to be organic rich peats or soils, that couldn't have formed any lower than mean high water. And so we provided the transfer function with these informative priors to then guide the reconstructions. To look at whether or not that actually works involves then validating the transfer function. So looking at the transfer function's ability to predict known elevations. And that's what you see here on this slide with the predicted versus the observed plot. I've also shown for you here the residuals versus the observed plot. And again, these elevations are in Swilly values. So again, a Swilly value of 100 being equal to about mean tide level, and then 200 being equal to mean higher high water. And what we found is that the relationship between predicted versus known elevation was good. So we felt really confident with the validation of the transfer function. So I'll move on to talking about what those reconstructed elevations look like. Here I'll describe for you in detail the reconstructed land level from the 1700 earthquake. And so here in the upper 50 centimeters of the core, we captured evidence of that substance in the form of this muddy peat soil identified here as a soil Y that is overlain by what we believe to be a tsunami sand. And that tsunami sand gradually transitions into mud. And then looking at the fossil diatom assemblages that we observed in the sediment, summarized for you here by their salinity preferences. So what we find then is that within this muddy peat, the soil Y, at least half of the diatoms found in that unit were identified to have a freshwater preference. But as we moved across that contact into the mud, we see a shift in the salinity preferences of these fossil diatom assemblages to a more brackish and marine preference. The Bayesian diatom transfer function then produced estimates of paleo elevation that you see here with the circles to 1 sigma uncertainty shown here with the whiskers. And what we find then is that the Bayesian transfer function did show that there was a sudden drop in elevation concurrent with that contact. And that the filled circles show you then that we have good modern analogs. And so these reconstructed paleo elevations were then reliable. And so that difference in elevation that we observed is a change in elevation of about 1.41 meters plus or minus 0.78 meters. And so then work that I'm continuing to work on and work that I hope to continue to do is to continue to apply this diatom transfer function to look at other contacts. As you can see here, the reconstructed elevations of the penultimate earthquake from the same sediment core, and then to continue to improve these modern analogs that we're seeing to really understand what's going on with the modern diatoms that we see along the Cascadia subduction zone, and then to build more of these estimates that we can then use to better understand earthquake hazards. And so with that, I would like to acknowledge a long list of collaborators who've helped in the field and or lab and or with modeling, and especially definitely with general discussions with diatoms, with earthquakes and otherwise. And that funding for this research could not have been done without funding from the National Science Foundation and research specific funds provided to me by Rutgers University. And so in conclusion, the modern diatom distributions that we observed provided a really important data set that could then be used to reconstruct relative sea level at Willapa Bay, and that this new diatom Bayesian transfer function was able to provide a reliable estimate of this land level substance from the most recent Cascadia subduction zone earthquake. And that future work will hopefully continue to improve the diatom transfer function and then try to produce additional estimates of land level change along the Cascadia subduction zone. So with that, I would like to thank you all for your attention, and I'm happy to answer any questions.
All right. Thank you very much, Isabel. Everybody give her a round of applause in the chat. I'll give you one right here. Thank you. It was a very, very cool talk. I always love to see the application of modern and paleo studies. So yeah, if anybody has a question, feel free to raise your hand or type it in the chat and we'll get to it. I'm going to just note a comment from Sarah about yikes of wading in the tidal muds. Did you ever get stuck out there?
Yeah, I absolutely did. That picture of me was actually my colleagues making fun of me because I ended up getting stuck to my waist and I had to sort of army crawl my way out. And so, yeah, it can definitely happen, but definitely safety first.
Oh, no. I wanted to ask you about the variability and species distributions within each of the groups. I noticed, and you commented that the least amount of variability were those diatoms and the shallowest of habitats. And I'm curious what your thoughts are on the influence of that, whether or not maybe hydrology or macrophytes resistance, prevented the distribution of those diatoms or the intrusion of diatoms into those shallower habitats.
I guess sort of to clarify, do you mean the narrow elevation tolerances of the freshwater species?
Correct, yes.
Yeah. I think honestly, part of that is just because working in these intertidal zones, the saltwater influence is going to be pretty broad. And so when we get to a point where we're kind of hitting the upper limits of tidal influence, I think that's the reason why we tend to see the narrowest tolerances of these freshwater species, just because they don't really want to creep any lower. And so part of that was sort of sampling limitations that I created in not really trying to trek too much more up into the upland environments where I think we would maybe see a wider tolerance, but the application of that for this type of land level reconstruction study is great because the freshwater environment is so narrow that if we're capturing that in the land level reconstruction studies, then our uncertainties are much smaller. And so I think that's a really cool application of what freshwater diatom species can tell us in this type of reconstruction study.
Great, thank you. I'm going to ask a quick question from the chat from Tom, and Tom wanted to know what diatoms were present in the core but not in the modern assemblages.
Oh, quite a few, Tom. Yeah, that's a great question. So one actually that jumps out to me is Cosmonace pusilla, which has been observed in other modern environments in Washington state, but for some reason I didn't really capture that in high abundance in my modern diatom distribution, but it was very much in high abundance in the fossil record. And so that's something that I'm trying to currently rectify in collecting more modern diatoms from places along Washington's coast that I know people have observed that species.
Thank you. Let's jump to a question from Sarah real quick. She's raised her hand, and then we'll come back and ask Sylvia's question.
Thanks, David, and thanks, Isabel, for a really fascinating talk. I think this is just such interesting topic, and I was particularly interested by the way that you are able to set your sea level boundary and measure the sort of ups and downs from that point. And I was wondering about, though, even like paleo events that can disturb the earlier events. For example, I mean, you saw the record of the 1700 event. How do you know that there have not been, I mean, we know that one for its recent time, but that there isn't disturbance by really big events of the things that are in that narrow set of record? Did that make sense?
I'm not sure. I'm not sure. Do you mean like an earthquake disturbing what was previously preserved there? Or
yes.
Okay.
Yeah.
Yeah. So what we do tend to know, even though we don't have a complete picture of Cascadia, what we do know from the paleo record is that we think that recurrence interval can be anywhere from 300 to 500 years. And that amount of time is certainly enough with the amount of sediment output that we're seeing in places like Willapa Bay, but also other salt marsh environments along the Cascadia subduction zone, that amount of time is enough to preserve the sediment record and to preserve the fossil diatom assemblages that we're seeing, because concurrent with that amount of sediment input is we have sea level generally rising in these areas. And so that's allowing for accommodation space that's going to preserve that sediment record on top of each other. So we're not necessarily concerned about the erasure of that record.
Okay, great. Thank you so much.
Yeah, thank you, Sarah. All right, so sorry to hop over you, Sylvia, but Sylvia asked the question, did your modern samples come from various substrates, or did you target one substrate across the habitats, for example, the surface sediment? Were you only sampling the sediment?
Yes, we were only sampling the sediment because I knew that ultimately the goal of the modern diatom study would be to try to reconstruct this land level change. I was interested in the diatoms that would be attaching themselves to the sediments that we saw preserved and not necessarily the epiphytic diatom species, for example. So that was definitely the goal, was to look at the benthic diatoms attached to the surface sediments.
Great. And I have another question in the chat from Tom, but first Mark has his hand up, so I'm going to call on you, Mark.
Okay. Yeah. Thanks, David, and thanks, Isabel. That was a really interesting talk. The question, I just have a couple tidbit questions for you. Did you in your training set, did you include the entire diatom assemblage, living and dead?
I did, yes, because I was unable to dye the diatom species to look at the difference between living and dead species. So it is a cumulative of the diatom species I counted were cumulative of what we saw on the surface sediment.
Okay, and was the sediment core site that Atwater and Hempel Haley worked on, was that in what's your bay, Willapa Bay? Was that from Willapa Bay?
Yes, sorry. Yeah, I got too excited to talk in talking about earthquakes to say that explicitly. Yes, the sediment core was taken from Willapa Bay upstream of the Neowicum River, which was one of the rivers where we sampled for modern diatoms.
Okay, and that core, you used dating models from that core from their study, or was the core dated? Did you date the core at all?
No, we haven't been able to. I haven't been able to date the core. I did send in some ages, but I don't think it was very good material. And so currently the stratigraphic correlations are what we're using to rely on the chronology established by Atwater and Hempel Haley. So yeah. Okay.
Just one last follow-up question. I'm always intrigued by sort of these transfer functions and what their specificity is. Is the transfer function that you've developed for earthquakes subsidence reconstruction, is it applicable beyond Willapa Bay?
Yeah, there's a lot of evidence to suggest that it will be applicable beyond Willapa Bay. And so one of the last slides that I showed for you, we have the reconstruction of the penultimate earthquake, and the subsidence contact across that event showed that there were also good modern analogs there. And so from a temporal standpoint, if we're able to go back a couple of thousand years and still see good modern analogs, I think there's a lot to suggest that it would be pretty applicable to the entirety of the Cascadia subduction zone, that a lot of those species that we're seeing are going to be useful in reconstructing land level change across the Cascadia subduction zone. And so that's something that I'm currently working on testing now. And then, yeah, future goals would be maybe seeing if we can apply that to other subduction zones.
Thank you.
Yeah.
Thanks, Mark, for those questions. Tom wanted to ask, was there any evidence of preferential preservation of diatom species, for instance, were you only seeing heavily silicified or lightly silicified diatoms?
That's a tricky question to answer because the absence of data doesn't really necessarily definitively tell us anything. But what I can tell you is that in looking at the slides, there was no evidence to suggest that these valves were undergoing any type of erosion or weakening, that the valves were almost always fully intact. They weren't broken. And so I think based on what I could see there, I don't think there's anything preferential as far as preservation bias is concerned.
Yeah, thank you. You had a great presentation and beautiful graphics.
Thank you.
Isabel, I wanted to ask you really quick because I heard a little bit about your background, and I know we get these questions a lot. Am I correct in assuming that you kind of started doing diatoms in a lab that wasn't doing diatoms before? You
would be correct, yes.
Okay, great. I share the same kind of history. For my master's work, I started doing diatoms in a place, and so I understand a bit of the curve that you probably went through to go through this. Now, some of the feedback that we got, and we spoke about this at the beginning of the talk, is that we're always hearing from marine folks of wanting to know more about marine diatoms. And I was curious if you could speak to any of the references or folks you talked to that were helpful in understanding the ecology or the identities of some of your diatoms.
Gosh, it's a bit of a chicken and egg problem, actually, because I tended to rely a lot on diatomists who are working in marine environments and using their studies and their plates to actually inform the identifications that I was using. So unfortunately, that's not a very satisfying answer for you, but I hope that that encourages other people to do similar work, and we can sort of maybe start to get bit of momentum going to create a stronger database of marine diatom species.
Sure, and just to clarify, the plates and such you were pulling from, were they more in like the primary literature or books or?
Primary literature.
Okay,
yeah.
Cool. All right, thank you. Sylvia has another question for you. How might sea level rise and other climate-related changes influence the future diatom composition of Willapa Bay?
Yeah. That's the big question mark, because what we did notice from just an ecological standpoint is that we began to see invasive species starting to come in, and that was also reflected with climate change as well, was also reflected in sort of the shellfish industry. That's actually really where a lot of great oysters are cultivated, is also in Willapa Bay, and the shellfish industry has been in a little bit of concern recently with the way that climate change is affecting these environments at Willapa Bay. So we do know that there have been changes happening as far as from the marine signature, and whether or not that's going to be reflected in these coastal environments is something that I'm interested in doing these sort of long-term studies and trying to look at what that change is going to look like with the diatom assemblages. So it's definitely something I have my eye on, and I think going back on a yearly basis or every few years would be a great kind of a study to do.
Great. Well, thank you. I'll keep my eye out for any last questions, but we'll start to wrap up things here. And I just wanted to say thank you again for a wonderful, multifaceted presentation on a multifaceted study. I think it's really exciting to hear more from the marine environment, and it's really great because I think you are the third marine talk that we are putting up in our series online. So thank you very much. Here we go. We have a question from, Sylvia just said, also thank you very much for explaining how you developed the Bayesian transfer function. And I don't think many folks have been doing Bayesian transfer functions, so that's great to hear about. Just as a reminder, next week, 1 week from today, Nick Schulte will be talking with us about developing multi-metric indices for applying at state level management using diatoms. And thank you again, Isabel. Really appreciate it. A lot of great shout outs to you in the chat. Take a look at that. And thank you very much.
Thank you. And thank you all for having me today.
Thanks everyone for joining. See you next time.
Bye.